So our next topic is neuroglia. What is neuroglia? Neuroglia are the non-neuronal cells in the central nervous system, that is brain and the spinal cord, and the peripheral nervous system that do not produce the electrical impulses. They maintain the homeostasis from the myelin and provide the supports and the protection for the neurons. So, in details, that is arise from word glia or the glial cells or the neuroglia that are non-neuronal cells in the central nervous system. In the central nervous system, glial cells includes oligodendrocytes, astrocytes, ependymal cells, and microglia. Okay? And in the peripheral nervous system cells, glial cells include Schwann cells and satellite cells. There are four main functions of them. What are the functions of them? Their main functions is to surround the neuron and to hold them in the place, to supply the nutrients and oxygens to the neuron, to insulate one neuron from another, and to destroy the pathogens and remove the dead neurons. So these are the four functions of them. And after that, they also play a role in the neurotransmission and synaptic connections and the physiological processes like the breathing, respiration type. Okay. Glial cells have far more cellular diversity and functions than the neurons, and the glial cells can respond to and manipulate neurotransmission in many ways. Additionally, they can affect both the preservation and consolidation of the memories. What is a glia? Glia was discovered in 1856 by the pathologist Rudolf Virchow in his search that is for the connective tissue in the brain. The term derives from the Greek word glue and suggests that original impression that they were the glue of the nervous system. And after that, there comes a type of the glial or the neuroglias, that is astrocytes. And what are the astrocytes? They are the most abundant types of the microglial cells in the central nervous system. That is the singular astroglia and plural astrocytes that have numerous projections that links the neuron together to do the blood supply and form the blood brain barrier. They regulate the external chemical environment of the neurons by removing the excess potassium ions and recycling neurotransmitters released during the synaptic transmission. Astrocytes may regulate vasoconstriction and vasodilation by producing substances such as arachidonic acids and the metabolites as the vasoactives. Astrocytes signal each other using the ATP, the gap junctions जिसको हम कहते हैं synapses between the astrocytes allow the messengers molecules IP3 to diffuse from the astrocyte to another. There are two types of astrocytes, protoplasmic and the fibrous, similar in the function but distinct in the morphology and the distribution. So protoplasmic astrocytes have short, thick, highly branched processes and are typically found in the gray matter. Fibrous astrocytes have long, thin, less branched processes and are more commonly found in the white matter. It has recently been shown that the astrocytes activity is linked to the blood flow in the brain, and that is what it is actually being measured in the fMRI. MRI is magnetic resonance imaging. They also have been involved in the neuronal circuits playing an inhibitory role after sensing changes in the extracellular calcium. These are subtypes of the glial cells in the central nervous system, and they are also known as the astrocytic glial cells. And star-shaped, there are many processes involved synapses made by the neuron. In humans, a single astrocyte cell can interact with up to 2 million synapses at a time at the free. There are foot processes. These foot processes forms the sleeves around the blood vessels together and make a blood barrier. You can see it is star-shaped neuroglia in the slide number 6, and it connects the neurons, many neurons. Oligodendrocytes are a type of large glial cells found in the central nervous system. Oligodendrocytes produce the myelin sheath insulating the neuronal axons, analogs to the Schwann cells in the peripheral nervous system. Some oligodendrocytes are also called satellite oligodendrocytes that are not involved in the myelination. ये कहाँ पाए जाते हैं? They are found only in the central nervous system, which comprises the brain and the spinal cord. And these cells were originally thought to have been produced in the ventral neural tube. However, research now shows oligodendrocytes originate from the ventral ventricular zone of the embryonic spinal cord and possibly have some concentrations in the forebrain. They are the last cell type to be generated in the central nervous system. Oligodendrocytes are the cells that coat the axons in the central nervous system and with their cell membrane forming a specialized membrane differentiation called the myelin and producing the myelin sheath. The myelin sheath provides the insulation to the axons that allows the electrical signals to propagate more efficiently. You can see in the image oligodendrocytes are present. There is some nucleus and there are nodes of Ranvier in the axon, dendrite axon, and it is connecting two axons from their myelinated portion. So myelinated sheet is connected to the oligodendrocytes and then it is connected to the other axon. एपेंडाइमल सेल्स क्या होते हैं? एपेंडाइमा क्या है? एपेंडाइमा बना हुआ एपेंडाइमल सेल्स है जिनको कहते हैं एपेंडाइमोसाइट्स, अ टाइप ऑफ अ ग्लाइल सेल्स. ये सेल्स क्या करते हैं? लाइन करते हैं वेंट्रिकल्स ऑफ द ब्रेन को एंड द सेंट्रल कैनाल ऑफ द स्पाइनल कॉर्ड which becomes filled with the cerebrospinal fluid, the CSF. These are the nervous tissue cells with a simple columnar shape, much like that of some mucosal epithelial cells. Early monociliated ependymal cells are differentiated to multiciliated ependymal cells for their function in circulating cerebrospinal fluid, that is CSF. Ependymal cells क्या होते हैं? These cells are involved in the creation and secretion of the cerebrospinal fluid. CSF बना रहे हैं and beat their cilia to help circulate the CSF and make the blood CSF barrier. They are also thought to act as neural stem cells. Microglia. माइक्रोग्लिया क्या है? न्यूरोग्लिया की एक टाइप है जो कि कहाँ मौजूद होती है? ब्रेन एंड द स्पाइनल कॉर्ड. Microglia accounts for 10 to 15% of all cells found within the brain. As the resident macrophage cells, they act as the first and the main form of active immune defense in the central nervous system. Microglia are the specialized macrophages capable of phagocytosis that protect the neurons of the central nervous system. They are derived from the earliest waves of the mononuclear cells that originate in the yolk sac, blood island, and early in the development and colonize the brain shortly after the neural precursors begin to differentiate. These cells are found in all the regions of the brain and the spinal cord. Microglial cells are small relative to microglial cells. Macroglial cells and microglial cells are different cells have the changing shapes and the oblong nuclei. They are mobile within the brain and the multiply within the different when the brain is damaged. They multiply when the brain is damaged in the healthy central nervous system. Microglia processes constantly samples all aspects of their environment neuron, microglia, blood vessels. In a healthy brain, microglia direct the immune response to brain damage and play an important role in the inflammation that accompanies the brain the damage. Minor diseases and disorders are associated with the deficient microglia such as Alzheimer's disease Parkinson's disease and ALS Neurons reach maturity a few weeks after the birth and cannot be replaced, as we know that neurons cannot divide after they are made, after they are grown. Damage to the neurons can either lead to the rapid necrosis and sudden acute functional failure, or it may cause to the death. These changes associated with the hypoxia, anoxia, nutritional deficiencies, infections, aging, hypoglycemia, poisons, trauma. So what happened? The axons of the peripheral nerves can sometimes regenerate if the cell body remains intact. Distal to the damage, the axon and the myelin sheath disintegrate are removed by the macrophages, the muscle supplied by the damaged fiber. In the absence of the nerve stimulation, the neuremia then regenerates about 1.5 millimeter per day from the joint of the injury towards the effector along its original track. And provided the two parts of the neuremia are in close positions, new Schwann cells develop within the neuremia, providing a pathway within which the axon can regenerate. Restoration of the functions depends on the re-establishment of the satisfactory neural connections with the affected organ. When the neuremia is out of the position or destroyed, the sprouting axons in the Schwann cells from a tumor-like clusters, traumatic neuroma, producing nerve pain, severe pain, and following some fractures and amputations of the limbs. And what happened to the neuroglial damage? When the neuroglial damage happens, as to the astrocytes, to the oligodendrocytes, when these cells are damaged, their processes multiply, forming a mesh or a scar, which is thought to inhibit the regrowth of the damaged central nervous system. Oligodendrocytes क्या करते हैं? Oligodendrocytes जब damage हो जाते हैं, these cells increase in the number and around degenerating neurons and also destroyed in demyelinating disease such as multiple sclerosis. In microglia, when there is inflammation and cell destruction, the microglia increase in size and becomes phagocytic, and they act in the pathogenicity, in the immunization, in the work there to produce some effects, the immune effects in the brain.